next video. All right, so the actual mounting of the specimen. You can see we have the slide here. I've put it on the staining rack because I need one hand to film. You will not be using the staining rack to mount your specimen. You'll hold the slide in one hand, you'll do all this on the other, or you can put it on the bench or whatever. It's not a big deal, on a paper towel, but don't use the staining rack for preparing your specimen. You have two hands, use them. All right, you have your culture here. Before you start, make sure the cap is loosened but not off. You have your loop sterilizer and your loop. Okay, to begin with, the first thing you do is, that's right, turn your slide over. We want to see what we've written. We don't want it washed off by the alcohol. So this is the actual surface that you will be applying your specimen to, the side that does not have the writing on it. Cool? All right. So what we want, first you want to make sure that you have a clean loop. It's not a bad idea to wipe it off a bit, maybe even get a little bit of water on a paper towel, wipe it to clean it, so that you're not putting weird, just random debris on your slide. Then you take it in and you sterilize it in your loop sterilizer. This is to sort of incinerate anything off that is on there, and also, of course, to make it sterile, because we are going into a live culture that we don't want to contaminate in case we, well, are going back and looking. Remember, we are looking for just these cells. Okay, so while you're sterilizing, go ahead and give this a good swirl. You can see these cultures have a bit of that sediment down in there. You want to mix things up so you're getting a good mix of the different cells that are inside. Once you've done that, just like before, you grab the cap, you take your loop in, get a good drop of it, and then you dab that onto your slide in the circle and just spread it around, okay? So you may or may not have seen, or you can see there's some moisture there, right? That's your specimen. It is absolutely full of bacteria. So don't feel shy about spreading it out. Once you're done, sterilize your loop. Put it back. All right, now what you need to do is you need to wait for this to dry. Dry fully before you heat fix it. You don't want to boil the stuff off. You want just to actually bake it into place. So we'll come back once it's dry. Fortunately, it being winter, that doesn't take very long. All right, so now at this point, you take your slide over to heat fix it. Okay, heat fixing, like I said, is basically baking it onto the slide, just like when you're baking with a glass casserole dish. Stuff sticks to the dish. That's what we're wanting. We're wanting this stuff to stick to the slide, but we don't want to incinerate it, right? So we have our hot plate. I have this set to 400. 350 to 400 is usually what we use. If you're using a hot plate that doesn't have a temperature adjustment on it, then set it to 7. Anyway, stick it on there, leave it there for maybe 5 seconds. That's all you need. And take it off. It might be hot. It's a good idea to use forceps. Don't leave it on there too long. You really shouldn't need to leave it on long enough that the whole slide will heat up and be hot to the touch, but it's up to you. Once you're done, then put it on your staining rack. Okay, so staining rack, here it's set up so that you can add the stain, and if it runs off, it drops into this tray, which has a little bit of water in it to dilute it so that it doesn't actually stain the bottom of the tray, which has happened here because it was used without water. Okay, we'll go through the actual gram staining process.